kind of been interested in diffusion and flow since about mid-80s.
Wow.
Yes, this is just kind of giving an this is a review talk. There's nothing, it's a little bit more of physics and how to think about diffusion rather than how to use it clinically. And this book of Derek Jones is kind of a large book. It's like a textbook. There are chapters there, but written by different authors. And I think anybody who is seriously interested in diffusion should have a look at it because it is chapters from methods to clinical applications and history. And look all over it. I think the behind, see, although diffusion has been known ever since the start of nuclear magnetic resonance, on which MRI is based, since about mid-1950s, early 1950s. Just after about 6 or 7 years after NMR was discovered, people noticed that motion, diffusion, flow, and NMR signal is sensitive to all these three things. So the fact that you can, the NMR signal is sensitive to diffusion has been known for a long time, say for 1953, for that, since about that time. In the mid-1980s, 1985, the Caprihan did some experiments, and he knew about diffusion. He knew that chemists and physicists had been using diffusion to look at cell size and to look at other materials and things like that. And then he thought, why not apply it to biology? And he was able to make diffusion images of the brain. And in about 1989, pictures like these came out, which showed that when there was cerebral ischemia in the acute stage, say within a few hours, 6 hours, or 3 hours after that, the image, wherever part of the brain that is ischemic, would become very bright in a diffusion weighted image. And this was the time when noticing images like these, people got really interested in seeing how could diffusion be used clinically. I'm not sure, I didn't show you, I should have shown you in MRI. One nice thing about MRI is that we can generate images with different contrast. This is a diffusion weighted image. I could have shown you a T2 weighted image, a T1 weighted image, and in those images, this sharp brightness, the sharp contrast that you see in a diffusion weighted image, indicating the location of infarct, would not have been that clearly visible. So this was a remarkable image. And this was this thing happens at acute stroke. When you wait over time, in days, this thing disappears. So so people started building models or trying to understand biology. What was this? What does this do? At about the same time, people found that tumors were also sensitive to diffusion variability. The location of the tumor, the images would appear bright. And although I have not talked about what is an ADC, but this ADC is related to this diffusion weighted image, and the region would appear dark. But even the tissue around the tumor would be affected. So this dark region here is an indicator that the diffusion is reduced. Here, and this region here is an example where the diffusion is increased. So the tissue around the tumor was also affected by diffusion. So there was. Not a lot of work has been done in this area because people are excited with the possibility that maybe the brightness here would be an indicator whether the tumor was malignant or not malignant. And it also tells you a property, tell you what's happening around the tumor and give an idea of whether the tumor was growing or not growing. because clearly the tissue surrounding the tumor was also affected by the tumor itself. And so people started thinking about what could be causing these things. And I think that's one of the things that we'll get into a little bit later on. Why does an image can appear bright like this? On the other hand, an image can appear dark because of reduced ADC. And then later on, the diffusion, people also noticed that diffusion the diffusion imaging can look at the anisotropy of diffusion, and white matter tracks, which are axon bundles, are anisotropic. I mean, are long thin tubes, and people could make measurements of FA, the direction in which the diffusion was maximum, and do some streamline type mapping, and construct pretty pictures like these. A lot of work at MRN is related with FA maps and tractography, because this is related to connectivity measurements between different parts of the brain and other such fun things. So in this talk, I'm not going to really talk about tractography. So the focus will be more on interpreting more basic diffusion mechanisms. So I think the first thing to understand slightly is what is diffusion. So diffusion is a random motion of particles by which matter is transported from one region to another region. So you can see there can be a number of particles. There can be a number of particles diffusing from this origin, and you can see the paths are all over the directions. And so there is, if you look at the particles, the mean motion of the particles need not be present. And the diffusion, what the diffusion is measuring is the mean square displacement of the particles over a given time. And this is the equation that Einstein was one of the first people to kind of be able to mathematically characterize this Brownian motion, and he proposed this equation. And so here we know that the particles, we have removed the mean flow. It is the displacement of a particle relative to where the particle was initially located. So there is no, in this definition of diffusion, there is no mean flow. Although mean flow could be present, but when you define the diffusion, the mean flow has been removed. And you can also see this TD, which is diffusion time. The longer the diffusion time, the diffusive particles will probe a large part of your region. So if the diffusion time is small, they won't probe that much. Okay, so, but this is the basic equation for diffusion. It's a random motion, and we are not considering in diffusion how blood is flowing from one region to another region, but it does have transport. So diffusion of water is about 2.3 millimeters squared per second. Diffusion of water in brain is 1 to 10 raised to minus 3. I'm figuring a decimal here. 1 into 10 raised to minus 3 millimeters squared per second. So what this means is that in 50 milliseconds, which is 50 milliseconds is the diffusion time, which is approximately the diffusion time in our MRI experiment. The particles will, or 2/3 of the particle will have a displacement of less than 10 microns. So when you do an MRI experiment with a 50 milliseconds diffusion time, you are only probing a region which is about 10 micron in diameter. You're not probing things beyond that. So that is one thing to remember. But the interesting thing is that, so although we are sensitive to this microscopic probing, but we are making MRI images at millimeter square, in a range of millimeters. Our Voxel size typically at 2 millimeter cube. So which is kind of fascinating that we can infer properties at macroscopic level, making macroscopic measurements. But so what we are measuring are statistical averages of these microscopic properties. we cannot follow an individual molecule, but over a large voxel, we can say what the average properties of what the molecules are doing. We can make those measurements. So here I'm trying to introduce anisotropy. So if you have a cylinder like this, and if a particle starts from here, what you can see is that the particle in a given time, if you look at its displacement in the y direction, it is more than the displacement in the x direction. So although because of temperature, the particle is moving at the same self-diffusion coefficient, but because of the restrictions that the tube provides, the particles can only diffuse more along the axon or along this tube than perpendicular to it. So in white matter, which consists of a bundle of axons, which can be in a simple way thought of like a collection of tube structures, there will be more diffusion or more sort of a mean square displacement along the axons than a mean square displacement in a direction perpendicular to the axons. So the diffusion coefficient we measure will be greater in the y direction than in the x direction. And that is the cause, I mean, that the fact that we can do that, we can measure this in anisotropy, has made diffusion imaging very useful in looking at white matter structure. So kind of want, before we start looking at how to interpret diffusion measures, it's kind of good to know what the NMR experiment consists of. We apply RF pulse. These things in red, these bipolar gradient pulses, are what tag diffusion. They tag, actually they don't, I mean, they will tag anything that is moving. They will tag something that is like a blood moving in an artery, which is moving with a constant velocity. They will tag things that are randomly moving. If you shake your head, they will also tag that motion. So these are motion tagging pulses. And after that, we do an EPI image. So the same experiment is used for measuring velocities. So if you want to measure a velocity in the aorta, you can use the same experiment to make velocity measurements. But for this talk, we are interested only in the diffusion. So the diffusion signal is given by some constant times some factor B times the diffusion coefficient, which diffusion coefficient, we remember, is kind of related to the mean square displacement. So what we are really measuring is the mean square displacement in this time, delta. And the B is an experimental parameter. In an MRI machine, I can dial in B. I can make B 1,000, whatever number, but it basically depends on the height of the gradient, how strong gradients are available, and then how long do I apply the gradients, and how far apart are these gradient pulses. So all the information that we derive are from this signal. And one thing about gradients is that the sensitivity of this diffusion depends on the direction of the gradients. So like previously we saw, if I apply the diffusion gradient in x direction, I will measure the mean square displacement in the x direction. If I apply the gradient in the y direction, I will measure the mean square displacement in the y direction. And so I have a tool which I can use to study in which directions the particles are moving more and how the displacement is dependent on the local tissue structure. And what is cool is that I can make a whole brain image from here. So I can look at these displacements at every voxel of the image. I can look at white matter, gray matter, and at every voxel. As I mentioned before, I'm not going to show any pretty pictures of the diffusion, but I kind of want to more focus on what is happening when we do these measurements. So one critical time is the diffusion time. So here there are three examples of low diffusion time, with higher diffusion time, and still the highest diffusion time. And what you can see is that if this is my voxel, in this for the very short diffusion time, the particles have not probed my voxel. So here they have probed a little bit more, and here they have probed the voxel fully. So what I will measure from here will be different in each case. So which is a kind of a problem, because in an MRI experiment when we don't have the freedom to choose the diffusion time. The MRI experiment sets the diffusion time for us. I mean, so it kind of fills in whatever time is there, like diffusion. And so the interpretation of so we kind of ignore this variability. And so the fact that our signal depends on how we have done the experiment, we are not really measuring true diffusion. So what we are measuring is some apparent diffusion coefficient, which is a function of the tissue structure, and is a function of how we did the experiment. And so if we were looking at material studies, where supposing we're looking at, say, porous rocks or something like that, and we can image for long hours, and we can change the experiment and probe diffusion more carefully. But in the MRI experiment we do currently, we have no control over it. So what we are measuring are apparent diffusion coefficients. But in the following, I won't make this distinction, but I will just say that we are measuring diffusion coefficient, and it will also be oblivious of the fact that we have some unknown diffusion time, which is in the range of 50 milliseconds. So the most common diffusion measures are fractional anisotropy. And as I said before, with diffusion I can probe diffusion in different directions. I can measure some, it's called diffusion tensor. It's like a correlation matrix. It's a 3x3 positive semi-definite matrix, and I can experimentally I can measure D. Then using some math, I can value decomposition. I can find out in which direction the largest diffusion is present. So I can have the largest eigenvalues, the other two eigenvalues, and then I can calculate axial diffusivity, radial diffusivity, mean diffusivity, and which is axial diffusivity is the diffusion is the largest diffusivity, and by modeling you can see that it will be along the axon bundle, and radial diffusivity is the sort of the average diffusivity in other two directions. And fractional anisotropy is a measured diffusion, which is a measure of how different are these lambda 1, lambda 2, and lambda 3. If all of them are equal, fractional anisotropy will be 0. If lambda 1 is much different than lambda 2 and lambda 3, then fractional anisotropy will be 1. So these are the measures that are most commonly used in the experiments we do at MRN. We principally calculate these measures, although now some people are trying to calculate other measures, but these are the most common measures here in our lab here. So just to give an example of the structure, I mean, like this is an optic nerve, this is a transverse section, this is a longitudinal section, and you can see that there is a myelin sheath here, there is an extracellular space, there is an intracellular space, and this is a longitudinal section. You can see there will be more diffusion along this axis, while there will be perpendicular to this axis there will be reduced diffusion. So there will be an anisotropic essentially. And people go and build mathematical models based on these structures, which something look like these. I mean, they build a collection of tubes, enclose it in some impermeable material called, they call it model myelin, and then they can do a computer simulation to see the effect of how this structure will change the diffusion properties. And so a lot of work has been done in this area with these and other more complex models. So this is one, I think that this is kind of interesting here. So we have, as I said before, we measure FA, RD, and AD. So if you assume cylindrical symmetry, then I can define a ratio alpha equal to RD over AD, and then FA is totally defined in terms of this ratio. So what you can see from here is that as the RD or AD become more and more, become 1, then RD becomes equal to AD, or if all the diffusibilities are equal to each other, FA becomes 0. Otherwise, it's kind of almost a linear decreasing function of this ratio alpha. The correct formula is here. This curve here is a plot of this curve. Okay, so now when we do experiments, we find that FA can decrease, FA can increase. So the question arises, I mean, what are the conditions in this model which will make FA decrease or increase? And so from this picture that you can see that whenever RD is more different than AD, FA is higher. So if I reduce RD, I will be making AD and RD more different, and therefore FA will decrease. But this picture is kind of interesting for the following reason. That FA, supposing you are over here with FA is 0.025. Then all this region, FA is higher than 0.25. So in the direction of the arrow, RD has increased. Normally an increase in RD would indicate to you that FA has to decrease, but in this case an increase in RD leads to a decrease or an increase in FA. So anything in this quadrant below, if a point moves anywhere in this direction, the FA increases. So the conditions when FA can increase or decrease are not that clear. That is, this is kind of one of the problems of interpreting diffusion data, because we are trying to infer data of microscopic structures from microscopic measurements. So there are multiple reasons which can cause an observed change. So FA can increase by reduction in RD, which is very easy to see, but FA can also increase by an increase in RD, which is a little harder to see. Okay, but then the diffusion imaging started by Caprihan. He was really interested in trying to see how to use these kind of experiments to infer things about perfusion. Because as we saw before, diffusion is the measurement of mean square displacement. So if there is a structure of tissue structures with vessels, and if there's perfusion in these vessels, then perfusion also is mean square displacement. Because the capillaries will be randomly oriented, so when things move, you will again see a loss in signal. And so he has defined, in the initial stages, he has defined this thing called Intra Voxel Incoherent motion. So that is what this experiment is really measuring. And if you do these experiments, say from B values, from low B values, you don't see a straight line, but you see a little curvature. So that curvature is perfusion dependent. But here, typically now these days the values are used around 1000. And as you can see, the effect of perfusion is not present anymore, essentially because the D that corresponds to perfusion has a much higher value. So the value of B that by the time you increase the value of B, that perfusion is signal from the voxels which are saying perfusion, the signal is destroyed. The signal goes to 0. So this is kind of indicating that the situation that we are measuring is more complex, and we have to be, effects that we see are dependent on the D values they use. Similarly, when you go to high D values, another thing happens. So there's a thing which is almost linear, and if you start using values, say 1500 and above, you again see a curvature. So this curvature is kind of being, a lot of people are trying to interpret this curvature and trying to see what biological information can be derived from this diffusion which is not linear at higher values. Linear diffusion equation corresponds to free diffusion. There are no restrictions. Whenever there is this, and this is basically the linear diffusion is Gaussian diffusion, where there are no restrictions, the particles can move as far as they can. Whenever we have restrictions, the diffusion is no longer Gaussian. And this is another picture of the same idea to higher d values, and you can see that this is a log scale. So one diffusion coefficient would have corresponded to this one, but the thing which is off Gaussian diffusion is shown by this curvature. So the question is, what is happening here? So how would you model this data? So people have thought of two ways of doing it. One thing is they say it's a two-compartment model. Okay, so we are making macroscopic measurements, and in this large voxel that we have, there are a lot of cells present. There will be intracellular components, extracellular components. So in some components the diffusion will be slow, in other components the diffusion will be fast. So we will model the diffusion by two compartments. And another model that is being proposed to handle this thing is, if you look at this line, it's almost quadratic. So let us assume, let us make a model which will measure how different the diffusion is from being linear after taking a log by some parameter called kurtosis. How different diffusion is from being a Gaussian diffusion. So both these models can be used to fit this data. So what do you do? I mean, so how do you choose which model is better? So there are kind of at this level there are two things. If I were to use just this model, I would not have any means of inferring what the structure is. So while this model is giving me some insight into maybe there are two compartments, maybe there is some intracellular, extracellular compartment, so I can derive more information from this two-compartment model than this kurtosis model. So the type of model like kurtosis model are purely phenomenological, because you're given the data and you fit it by some convenient mathematical equation. While the two-compartment models are trying to go closer to, they're trying to say, okay, maybe let's try to make a model that will reflect biology of the tissue structure, and so maybe if you model the two-compartment model, we'll learn a little bit more. But having said that, I mean, people, when this is kind of obvious thing, so people are, so that there has been, these kurtosis models have been kind of pushed by the group by Halpern and Jensen, and Fearmans in their group has tried to interpret the kurtosis model in terms more of biological structure. But the general point I would like to make is that if you develop a model which relates to tissue structure, then it will potentially be more useful than some model which is purely mathematical. But if you are interested in something like classification, then you probably don't care, and then you would just try to get some parameter which represents the data and see if it's different in two groups or not. Okay, but there's another thing here. So now if you look at the two-compartment model, there are 1, 2, 3, 4 unknown parameters. And if you look at kurtosis model, there is 1, 2, and 3 unknown parameters. So it is a little easier to fit the kurtosis model. So when you're fitting your data, you have to not overfit what you observe. I mean, it is very time-consuming to obtain extra data points. And so the choice of the appropriate model also has to be considered from the point of view of overfitting or not overfitting, and also from whether it will lead to some useful biological information. So let us proceed further with the two-compartment model. So in the beginning, when it was proposed, there was a lot of excitement in saying that slow diffusion is intracellular, fast diffusion is extracellular. And people fit that diffusion model, and they found that the fraction of the fast diffusion was 0.67, the fraction of the slow diffusion was 0.33, and they could calculate these things. But then there was a problem. by independent measures, the extracellular volume fraction was coming in the range of 15% to 30%. While the model fitting of this two-compartment model to MRI data was giving a fraction of 0.6. So this model was not compatible, although we divided up the diffusion rate into slow and diffusion. The jump to intracellular and extracellular interpretation was not correct. And it did not fit what people were observing about extracellular fractions by other measurements. Even by looking at the electromicrograph, if you could calculate what the extracellular fraction was, it was not coming to 67%. So there was a problem. So people did MRI experiments which are focused on especially looking at extracellular volume or intracellular volume in cell suspensions. Even those experiments failed to prove that this thing is really reflecting biology. So there was again kind of a little difficulty in interpreting what is going on. So there are two compartments, there are slow diffusion and fast diffusion, but whether it is really corresponds to intracellular or extracellular was not clear. And in fact, during those times, Washoe group, there was a very good physicist there, Blonsky. He showed that even in a single compartment without restrictions, you can see slow diffusion and fast diffusion. And this was because of the fact that if you have a situation like this one, so here I've taken a point in the center, but there are particles at the edges. So the particle at the edges will see restriction, the walls. The particles in the middle are not seeing the walls. But when the diffusion time is small, there will be a discrepancy that the particles at the edges will see a partial restriction. The particle which will slow down the diffusion. The particles in the middle will see fast diffusion. And if you do an MR experiment, you will see two compartments. They're basically a exponential decay. And so it was shown theoretically and then later experimentally by a different group in Washington. Okay, so then the question arose, how should we modify the model to explain this? So that essentially that we have to admit the fact that extracellular volume is small. And so the idea that was proposed was that along the cell membrane there are other lipid layers which the particles can attach themselves to, and the diffusion is slow. And inside the cell, there is again fast diffusion. So basically, people moved on to assuming that there will be fast diffusion outside the cell as well as inside the cell. And the slow diffusion is caused by some lipid properties near the cell membrane. And then, at least with this thing, the discrepancy that we had for explaining the extracellular model was explained. So now also what's happened is the diffusion experiments are getting more and more complex. So the MR technology is improving. We can use higher and higher gradients, and we can use more and more gradient directions to probe diffusion. So there is an opportunity to build more complex models which will be closer to the tissue structure. So one model that has been proposed is which includes, say, so these dark grays are cells, this white is extracellular space, and there is some particle moving around. And so what the model is that inside the cells, they model it by restricted diffusion. So these cells are like rods. So this is the cross section. The diffusion will be restricted in this region. In the extracellular space, they model the diffusion by a tensor. They call it Hindered diffusion. And so normally when you do an MR experiment, there can also be CSF present. So in the CSF space, we assume there is isotropic diffusion. So the model is built, which consists of these three components, and then we can apply use the diffusion data to infer parameters of these models. So models of this nature were presented by Asaf from Technion in Israel, and then simplification of these models have been presented by pushed by Daniel Alexander, and he has termed this technique MODI. So going back to the original picture that we had, so how do you explain this brightness? So the explanation is the ischemia will lead to cell swelling because lack of oxygen will cause changes in osmotic balance and more water will go into cells. And the model we have for cell swelling is that the layer of the cell membrane will volume of the cell membrane of the layer near the cell membrane will increase, and we know this causes slow diffusion, and therefore the diffusion after swelling will, on average, will slow down, and which will lead to the signal decay to be less and the image to become brighter. So essentially, what we are saying is that fraction of Fs after cerebral ischemia, the fraction of Fs increases, and the fraction of the fast flowing component reduces. We are not saying that the diffusion coefficient changes. We are saying that the fraction of slow of the region where there was slow diffusion increases after cell swelling, causing this brightness. And what is observed is that when after a while this thing disappears, and that is caused by the cell membrane breaking down and the liquid leaking out, and becoming more like vasogenic edema or more in the extracellular space, or all the water in the extracellular space, and then the fast diffusion component increases, causing a decrease in this intensity. In tumors, the concept is of cell proliferation. So the tumor causes increased number of cells to get produced, which again causes an increase in the fraction of the slow-moving component, and the diffusion weighted image gets brighter, and or the diffusion coefficient increases in that area. And around the tissue there is vasogenic edema, which causes the diffusion to be faster and the signal intensity to go down. So now here, doing TBI work, Andy in his study, Andy and Joseph has seen that in their longitudinal study they found in people with traumatic brain injury there was increased effect. And they have listed a number of reasons which can cause that. So again, the problem is all these reasons can cause an increased effect, but if you do see an increased effect, we are not in a position to infer whether which was the specific cause. This is I think the problem with a lot of MRI when it's applied to biology, because the biology is so much more complex. We're making these microscopic measurements. I mean, we can give a reason which will cause the FA to decrease, FA to increase, but if FA does decrease, we are not sure what has caused it. So this is my summary. I mean, the key point what I want to make is that what we are dealing with is an inverse problem, so that there are multiple models that can fit the experimental data. So we have to take into account the biology and what we know from other prior knowledge or from other interpretation of biology to make an appropriate model. Biophysical models that are based on some tissue structure are probably more useful than something which is purely phenomenological. But more importantly, what's happening is that the MRI technology is improving. So we can use higher gradients and lower gradient directions, and we are in a position to probe diffusion at a greater complexity and actually build these models rather than just talk about them theoretically, which makes diffusion a very exciting area.